have him in this seminar series of liver toxicity and steatotic liver disease, which is it's like a continuation. It was a way of securing our cost action network that it was created during the cost action. So uh, mm, we are very pleased to have him with us. So please, Michael, please do introduce him
Yeah,
to
well,
us.
actually, you made the perfect introduction already. This is excellent. I mean, once again, it's very glad to have Volker with us, and once again, now recorded to thank you for accepting the invitation to participate in this seminar series, which actually, as Maribel explained, is an outcome of the cost action, and now in this EASL-DILI consortium. So, Volker, I think I don't give you another introduction. I mean, people can see your biography on the announcements, but once again, thank you very much for accepting, and please, the floor is yours.
Yeah, thank you very much, Michel, and everyone for the very kind invitation. It's really my pleasure, and looking forward to a nice dialogue then also thereafter. So today I would like to present a little bit our work on metabolic liver disease specifically, and how these events and pathologies can be modeled in a 3D environment in vitro. So a short background into our activities. So we come predominantly from the human tissue engineering background or context, and I think what sets our approach a bit apart there is that we are working exclusively with primary patient-derived samples. So this is not cell lines, it's not stem cell-derived tissues, it's not animal cells, so it's really all primary human cells isolated from organ donations or tissue resections. And we put a lot of emphasis on these integrative omics analysis, so which means that we are benchmarking cells directly after isolation as fresh tissue as possible, yeah, and then compare our 3D tissue models, always referring or relating back to these freshly isolated cells, in order to really understand which parts of the tissue our models are closely resembling, and also maybe which features might be somewhat lost. We've worked predominantly with human liver, and this will also be the main focus of today's work. But more recently, we've also branched out into other tissue models, adipose tissue, pancreas, skeletal muscle, and more recently also intestinal models and cancers. And I will give just a brief outlook in how we are integrating those tissue models to also understand crosstalk between the liver and other tissues in the context of metabolic disease. And then starting from normal healthy tissues, if one can call it like this, we've then also used patient-derived material to model various aspects of disease, metabolic diseases. Fatty liver is the scope of today's talk, but also inflammatory diseases and infectious diseases. We then integrate those different tissue models, as I already briefly foreshadowed, yeah, in microfluidic devices that we built in our own clean room facilities here at Karolinska, in which we can then study crosstalk between different tissues, and also how we can modulate this crosstalk using pharmacological interventions. And lastly, also for what we call here precision drug development. So since all of those cells are directly patient-derived, they recapitulate this inter-individual variability, and this means that already at very early stages in the drug discovery process, we can use these scalable tissue models for parsing inter-individual variability. So with this being said, first a rather seminal slide. This was our initial publication from 2016 in close collaboration with Magnus Ingemann Sundberg's group at the Karolinska, where I did my postdoctoral studies back in the day. So this shows the overall process of the formation of these 3D human liver spheroids. So essentially we start with isolated cells. These are cells that we release from human liver explants or resections using two-step collagenase perfusion techniques. This ends up with cell suspensions, and then we are seeding very well-defined numbers of cells into ultra-low attachment plates with a little U-shaped bottom, exactly 1,500 cells in each well. And then over the course of 7 days, the cells are sedimenting. And because the plates are cultured or treated in a specific way, that the cells cannot attach, the cells attach to each other and then form these round spheroidal aggregates that are not well connected to the plastic. Yeah. And on the right-hand side, this is the initial proteomic characterization. So here we did the experiments in close collaboration with our colleagues from the Department of Liver Transplantation. One part of the livers of individuals undergoing liver resections, we essentially immediately snap froze in the surgery room. And from the rest, we isolated the cells and then seeded them either in conventional 2D culture or in 3D culture. So each column here now is one sample, each row is one protein. And what I hope becomes clear is that the livers indicated here with the black dots and the 3D cultures indicated with the green dots, they form one cluster, whereas the 2D cultures of exactly the same individuals, isogenic 2D cultures, are fundamentally different already as early as 24 hours after culture. So this really means that it's the culture method itself that drives major phenotypic change of liver cells in a very rapid timescale, or on a very rapid timescale. The same is then represented here in this principal component analysis, where again livers in 3D culture form one cluster and 2D are fundamentally different. And when one compares liver and 3D culture from the same donor, they cluster more closely together than, let's say, the livers on one side and the 3D cultures on the other side, which again indicates that this inter-individual variability that exists between human livers and that can be quite substantial can be carried over at least to a considerable part into the in vitro context. So this was the initial benchmarking. We've then also done stability evaluation. So this is viability as measured by ATP concentrations as a proxy. And overall, these ATP values remain reasonably stable for many weeks. This is from the initial publication where we showed 5 weeks. We've now pushed the system even further to up to 16 weeks. This is not yet published. So it's a very long-term stable overall culture paradigm. We've also done metabolomics of those spheroids. So each dot now here in these scatter plots is one metabolite. Blue dots are endogenous metabolites. The red dots are exogenous metabolites that are metabolized from 5 different probe substrates for different cytochrome P450s. And overall, what I hope becomes clear is that both the intracellular metabolome as well as the extracellular secretome remains reasonably stable over at least 3 weeks in culture. So the abundances in freshly isolated cells is plotted on the X, and after 3 weeks in spheroid culture on the Y. And correlation coefficients are in both cases above 0.9. So this means that not only cytochrome P450 activities remain reasonably stable, and also phase 2 activities as indicated here by some sulfates, for instance, but also the endogenous metabolism, amino acid metabolism, carbohydrate metabolism, fatty acid metabolism as well. So this then opens possibilities to also study and manipulate endogenous metabolism in such 3D culture methods also in longer periods of time or in chronic exposures. So when it comes to applications, there is a multitude. Hepatotoxicity predictions is one and maybe the most obvious one. We've done quite a bit of work on those. Also pharmacokinetic analysis, so evaluating directly hepatic clearance of different compounds, drug-drug interactions, so how compounds are inducing various cytochrome P450s and how that might impact the clearance then of other victim drugs. As well as metabolite identification of direct human metabolites of a given compound of interest. And then, of course, also more basic studies of liver biology, how the different hepatic cell types are interacting, hepatocytes with various non-parenchymal cells, be that inflammatory cells, be that stellate cells or cholangiocytes, and also the modeling of a variety of different liver diseases. And this will be the focus of today's talk. So we've done quite a bit of work on muscled fatty liver disease, a little bit less or quite considerably less, I should say, on cholestatic models, and also viral hepatitis or viral infection models. And here maybe I could emphasize that we have a very good working relationship with the Swedish Public Health Agency. So in Stockholm, we have the only BSL-4 laboratory in Scandinavia, which is just across the street. And so we can deliver our liver spheroids and then infect them in a controlled BSL-4 environment with really nasty hepatotropic viruses, hemorrhagic fever viruses, Ebola, dengue fever, Crimean Congo hemorrhagic fever virus. So viruses that infect hepatocytes, but that are conventionally very, very difficult to study when it comes to viral entry factors or novel antivirals. So just in a recent study, we identified the viral entry factor for Crimean Congo hemorrhagic fever virus, which happens to be the LDL receptor. So now focus on muscle. I don't think I have to give much background here to this audience. Just to say that it's a umbrella of or different manifestations of fatty liver disease, starting with hepatic steatosis, which is a relatively benign intracellular lipid accumulation, which then deteriorates in a subset of individuals into more inflammatory conditions called MASH. As well as eventually end-stage liver diseases, again in a subset of those individuals, hepatic cirrhosis and HCC. And importantly, there's only a single treatment so far approved, resmetirom. I believe that was last year, March or so. But also resmetirom is only effective in around 25 to 30% of patients. Or resulted in improvements in only 25 to 30% of patients in the pivotal phase 3 randomized controlled trial. So with this being said, we started to then slightly modulate or modify the culture protocol that I briefly introduced. Briefly, I showed you spheroids using liver cells from healthy individuals. And now we use essentially the same culture method, but use cells directly from patients with a histologically manifest diagnosis of MASH as comparator. And so the first thing that we saw is when we use cells from MASH patients, we see that the spheroids are larger despite having the same cell number. So this already indicates some form of hypertrophy. Cells are more insulin resistant. This is a phospho-AKT staining after insulin stimulation. And when one looks at a confocal image, one nicely sees steatosis. So overall, this is one spheroid. Overall, the diameter is around 250 microns. This would be one hepatocyte here, and one can nicely see it's essentially jam-packed with small lipid droplets here. This would be microvesicular in its steatotic pattern, and essentially the only place in the cytosol that is, or the only place in the cell that is devoid of these little droplets is here in the center, which would be the nucleus. And other cells are already macrovesicular here in its steatotic pattern, so few but substantially larger droplets, and some cells maybe are even already ballooning. So this would be by imaging. We can also use a variety of different biochemical assays to really quantify the extent of hepatic steatosis in those 3D cultures. Doesn't stop at the level of steatosis. We can also measure pro-inflammatory cytokine secretion. We see that this is elevated when we use patient-derived cells. We also see that in patient-derived cells, there's more extensive collagen 1, A1 deposition, also increased secretion of pro-collagen 1. And maybe what is most interesting of this data was when we compared the overall transcriptomic signatures, where we used data from the literature. This is, I believe, a total around 300 MASH and controlled biopsy samples. This is literature material that we mined, and we compared that and cross-referenced it then with samples in total 20 different MASH spheroids and controlled spheroid samples, total transcriptomic analysis. And what I hope becomes clear from this principal component analysis is here multiple things. So first, not surprising, it's a very heterogeneous disease. We see a huge spread between the individual distributions of those dots. Nevertheless, there is also a clear difference in patterns between the biopsy samples, MASH and control. And when we now overlay this with the MASH and controlled spheroid samples, we see that this distribution is somewhat recapitulated. So MASH spheroid samples overlap with MASH biopsy samples, controlled spheroid samples overlap with controlled biopsy samples. And if we now collapse all of this data along the first principal component, we see that the density distribution is really reasonably similar, so peaking at essentially the same areas. So this means that despite this heterogeneity among the samples, we can nevertheless use spheroids as a model to also recapture this heterogeneity in an experimentally accessible in vitro context. One additional thing that I would like to point out is, of course, our spheroids don't only have hepatocytes in them, so they're mixtures of hepatocytes and various non-parenchymal cells. And here we zoomed into the immune cell repertoire. This is a relatively sophisticated gating strategy. We've done this together with Niklas Björkström at Karolinska, who was really an expert in liver immunology. And overall, what became very clear from this data is that in the immune cell repertoire that we have represented there, it's not only Kupffer cells, but it's really the entire portfolio of immune cells that are present in the human liver at the time of isolation. Not only classically defined liver resident immune cells, but also infiltrating immune cells, and also a variety of the different subsets. This can be donor, there can be substantial differences between the donors, but overall this allows us to also recapitulate events that might require different and individual subset populations of the immune cell repertoire. Previously I showed transcriptomics. We've also done other omics to first really understand what happens in our model at the comprehensive unbiased molecular level. So here on top, this shows a proteomic characterization at the pathway level. Again, upregulated in the mesh samples were pathways related to ECM organization, collagen formation, cholesterol biosynthesis, so pathways that one would essentially expect. Whereas downregulated were conventional or classical hepatic functions, glucuronidation, the complement system, biological oxidation, which is also including cytochrome P450s, and so on. So indicating some partial loss of hepatic or hepatocellular functionality, which also has been described in more fibrotic livers. We've also done lipidomic assessments, and here again, little surprises. So in the mesh samples, we see drastically overrepresented diacetyl and triacetyl glycerols, whereas the LPC and PC species are relatively upregulated in the control samples. And what was overall interesting is that when we looked at the individual pathways in RNA and protein level, then we saw that at the RNA level, we saw inflammation and fibrosis well reflected, whereas steatosis was exclusively detectable at the protein level. So this really means that if one wants to assess the molecular effects at the steatotic level, one either needs to, of course, evaluate metabolomic signatures and lipidomic profiles, or one needs to look at the protein level abundances, but at the RNA level, changes at fatty acid synthase, for instance, were not apparent. So with this, we were then first reasonably happy with the overall characterization, and were confident that the model would indeed carry over those key hallmarks of the disease, steatosis, inflammation, fibrosis, into the in vitro context. And then we used it for a variety of different applications in drug discovery and development. The first here was a screening of a defined chemical series of compounds that were involved in DNA damage repair and cellular bioenergetics. This we've done together with Maurice Michel. He's a very talented medicinal chemist at Karolinska, who has developed these organocatalytic switches that can alter directly enzymatic function of different candidate enzymes. And specifically, we zoomed in here into a molecule called OGG1. OGG1 is an enzyme that is involved in the removal of 8-oxoguanine. This is a DNA modification that results from excessive oxidative stress. And normally 8-oxoguanine is repaired or OGG1 removes or generates these aporinic sites. And these aporinic sites then are further excised by an enzyme called APE1, and then the gap is subsequently filled in by DNA polymerase to give rise to a fully repaired double-stranded DNA. And what now these organocatalytic switches are doing, they are binding to the active site of OGG1, acting there as a novel electron donor, and thereby provide the enzyme with a novel beta-delta lyase activity. And as a result, instead of generating an aporinic site, now this entirely excises already the backbone of the base and only leaves the phosphate. And this is then afterwards simply removed by phosphatase, thereby bypassing APE1, which is the rate-limiting step of this DNA repair pathway. And as a consequence, DNA damage repair is up to 20-fold faster. And this is a rather elegant approach because these organocatalytic switches, as they say, are really catalysts. So which means they are not metabolized in any way, but they can also be reused in the same enzymatic reaction. And therefore, they require very, very low concentrations in order to be biologically active. And when we did then our screen for antifibrotic molecules of this chemical series, we indeed found some OGG1 modulators as some of the top hits. One of them, then we zoomed in, this TH10785. We saw a dose-dependent reduction in procollagen secretion, and also consistent with the overall hypothesis, we saw that there was a reduction in these DNA damaged foci. So this is here a staining by p53 binding protein. There are many of these foci in the mesh samples, virtually none in the control samples. And if we treat those mesh samples then with TH10785, they are significantly less. So being in line with the facilitated DNA damage repair. So this nicely shows that for a proof of concept, we can zoom in or prioritize different compounds in the chemical series for the most effective ones. And another approach is chemogenomic profiling. So chemogenomics, very briefly as a concept, uses small molecules with a very well-characterized pharmacology. So they have low or high selectivity, low polypharmacology, are already very well characterized, low toxicity. They are cell permeable, and importantly, one exactly knows the target. And so there we are part of a large project called EUB Open, which is a large EMI-funded initiative that had the goal by the year 2025, so ending this year, to develop chemogenomic probes against 1,000 human proteins. And indeed, this target will also be met. And it's part of an even larger initiative called Target 2035, which has a goal by the year 2035 to have developed chemogenomic probes against every protein encoded in the human genome. So then you can imagine that if you have a phenotypic screen like ours with a mesh model, steatosis, inflammation, fibrosis, yeah, and you have the entire library of chemogenomic probes against every protein in the human genome, you can do a large screen, evaluate, okay, these 500 hits are now significantly reducing steatosis. And importantly, and this sets chemogenomic screening apart from normal screens, one can draw a direct line between the hit molecule and its actual target because the pharmacology is so well characterized. So this means one can directly, in a pharmacologically accurate way, associate any protein that is involved in a certain phenotype. And this is then a really nice way for systematic target identification. So we've done this here first in a pilot screen, in total 200 or so different probes that we tested that were ready at the time of conducting these experiments. It's a multiplex system. So from the spheroids, we image for steatosis using high throughput automatic confocal microscopy. And in the supernatants, we evaluate from the same spheroids inflammatory marker and fibrotic markers for procollagen secretion levels, as well as 3 different cytokines. And as a consequence, then we identified multiple antifibrotic, anti-inflammatory, and anti-steatotic hits. Most of them were inhibitors, also one activator. We were first happy to see that some of the usual suspects were indeed there in an entirely unbiased orthogonal fashion. So ALK4/5, known antifibrotic target, the TGF-beta receptor here, was again clear as an antifibrotic hit. BRD inhibitors were anti-inflammatory, so also those already entered the clinics. But then there were also many new compounds or new targets that were identified, which we then further zoomed into a subset of those, and essentially each one of them can form the starting point for now new drug development campaigns. And specifically excited we were about this muscarinic receptor TRPM8 axis. So very briefly, maybe I don't have to go into too much details here for the sake of time, but using a series of different BRED assays, we identified that 2 of those hit molecules, specifically PQCA and this PF compound here, were directly connected along a very simple molecular module. PQCA is an activator of the muscarinic receptor, and then we could show using these BRED assays, and if there's some discussion around that. I would be happy to go into more details also afterwards in the discussion. But we could demonstrate that PQCA activates the muscarinic receptor. Then this results in activation of the GQ transducer, which in turn activates PLC. And PLC metabolizes this PIP2 molecule, which happens to be a cofactor for TRPM8. So we see that we can phenocopy the inhibitory effect of this TRPM8 antagonist with an activation of the muscarinic receptor. And this results in an activation of quimodulin, and how this is then overall related to or linked to fibrosis, that remains a further subject of discussion and investigation. So it's an answer that I at present cannot give. But in any case, we were very excited that two molecules that were entirely independently identified based on these probes could be linked at a reasonably short module. And so now for those, we are currently investigating the effects in knockout mice on high-fat diet, whether those are indeed replicating effects in mesh models, and also have developed new compounds against the TRPM8 calcium ion channel that might also serve as more drug-like molecules, whereas those here are probe substances. They are not made for beneficial pharmacokinetics. So another example here, and this is very, very brief, essentially fresh off the press. So we've done a chemogenomic screen for the nuclear hormone receptor family. Again, steatosis, fibrosis, and inflammation. And we saw nice patterns of hits. So here, different modulators of the PPARs. So this is the NR1C family, are the PPARs. We see a reduction in steatosis, which nicely fits with the literature and expectations. On fibrosis, we see hits here for FXR agonists. Also, that is not entirely novel, so we were very happy to see a replication of this. But then we also see clusters of additional probe hits, which were much less expected. So here, for instance, antifibrotic effects, quite strong and significant antifibrotic effects of the RAR activators, so retinoic acid receptor activators, and antisteatotic effects of ROR. So this is something that we are now also looking into more mechanistic detail. Similar then for other compound here. The previous one was a premenopausal donor, female donor. This is a male donor. So given that the nuclear hormone receptor family also includes sex hormone receptors, therefore we screened premenopausal, postmenopausal, as well as male donors, which also links back to what I tried to already slightly foreshadow in the beginning, that one can screen here at the level of already inter-individual variability in responses. So we are validating some of the interesting hits. We're screening also other libraries, ion channel, GPCRs, and also are extending this to other tissue models, particularly adipose tissues is what is of primary interest for us at the moment. So, and now for the last 15 minutes or so of this talk, I would like to talk briefly about the integration of those 3D liver cultures, patient-derived liver culture with other tissue models and microfluidic devices. Because while it is clear that fatty liver is, of course, a predominant liver-specific manifestation, it is nevertheless integrated also within a context of overall metabolic dysregulation. And we've established those other tissue models here, adipose tissue, all again from primary human patient-derived material, and importantly, all in exactly the same media. So those it's exactly the same media composition, which allows a direct co-culture between those different models. So this would be one example. I will not go into any depth here, but these are all published. So this would be the adipose model. So they have very large cells, up to 50-60 micron each cell. So very huge cells. In vivo, they can be even larger. So maybe it's still not fully there yet. Also, those cells are metabolically activatable still. This is again a phospho-AKT staining. We have skeletal muscles which retain their striation. We can measure calcium release. We can measure force generation. We can still stimulate them electrophysiologically. And also pancreatic islets, again, fully intact human islets. They retain the functional composition, beta cells, alpha cells, delta cells. We now also know epsilon cells are still present. and they remain stimulatable by differences in glucose concentration. So if we expose those islets on chip with postprandial glucose levels, we see indeed also insulin secretion at physiologically relevant concentrations, whereas with fasting glucose levels, there's essentially no insulin secretion detectable. So then first, how do we fabricate those chips? We use a liquid polymer. So we essentially inject a monomer formulation into what is called a mold. So we inject this is indicated here in yellow, then shine on UV. The UV cures those monomers. Essentially, it hardens, it polymerizes the monomers. Only those parts that were exposed to the UV are then hardened out, and then we can disassemble this entire contraption. These sprues and runners here are not attached because they were not exposed to UV, so any monomer in those is still liquid. Yeah? And overall, the cycle time takes only around less than a minute, so essentially as long as watching the movie of it. And this means we can then very rapidly generate a replica of whatever mold structure we have, and then we have a new microfluidic chip or device. So this was the overall fabrication process. Now the next part, the next box to tick in a way is now the material. I already said that it's very convenient to have a liquid material because we can produce in these devices in batch. And normally those devices are, I would say, more than 90% in the literature are using PDMS. It's a liquid elastomer, has also some beneficial properties. But it's very notorious for its absorption of different compounds. And then there's also various other materials, anything from Teflon, polystyrene, COC, polycarbonate, polypropionate, and PMMA. And but nobody had done an actual direct cross comparison of the absorption of different compounds in those different materials. So what we've done here is we fabricated exactly the same devices, exactly the same designs, using those 8 different polymers, and then exposed those polymers to 6 different drugs with very different physicochemical properties. So essentially we have a defined solution, pipette that into the chip, either wait 2 hours or 24 hours, pipette it back out, put it on the mass spec, and see how much of the compound is actually left. And I must say, although PDMS was of course very notorious for its absorption already, I was still stunned by how much difference in absorption there actually was. So when we then ran our assays, I'm just showing here 3 of the compounds for simplicity. Those are the hydrophobic compounds, chlorpromazine, tamoxifen, and montelukast. So the lowest sample here is PDMS. So this means that after as little as 2 hours, 99.9% of the compound actually absorbed into the bulk polymer. So it's essentially one is working with virtually homeopathic concentrations. And the other polymers are all performing better, and eventually then we choose our thiolene epoxy material because it had the lowest absorption across all the different compounds. So more than 1,500-fold less. Well, absorption is still considerable actually. So it's still around 80% absorption, but 80% absorption is still at least substantially better than 99.9% absorption. And so this is in a cell-free context, and then we also evaluated that in the context of our liver spheroid assay. So we now add liver spheroids either to the thiolene epoxy chips or to PDMS, expose them here in this example to chlorpromazine again, the one with the worst absorption. And indeed, in line with what we measured previously by mass spec, we see no toxicity whatsoever when we use our PDMS devices. So even the highest concentration, no indication of toxicity whatsoever. And again, this is because 99% of the compound is not bioavailable anymore, whereas we see much higher toxicity at virtually relevant concentrations in our thiolene epoxy devices. And the same for tamoxifen. We also had hydrophilic compounds as a control, if one wants to call them like this, acetaminophen, for instance, and there's no difference whatsoever. So as long as one is working with hydrophilic compounds, the material essentially doesn't matter. It doesn't stick to anything. As soon as one works with hydrophobic compounds, log P below 3 around, then absorption is massive and really strongly benefits from being controlled by choosing the right material to work with. So this is the second part, the material aspect. Now the last is now the design. So we use here the device that is a pneumatically actuated chip. So let me briefly explain how that works. So it's essentially three different layers that are then assembled. Those central compartments here are the important ones. Blue would be the cell culture medium. We have one central chamber and two satellite chambers. So the different tissue models would be loaded into the satellite chambers. This is a incarnation of the device with 2 satellite chambers. We also have other designs with 4 satellite chambers. So here now we add, in the example that I will show, liver on one side, pancreas on the other side, islets on the other side. Yeah. Then we are closing the device and then starting our pneumatic actuation. So this means that the center of the chip is connected to a pump, can either be a syringe pump. Now we use a pressure controller. It's a little bit more sophisticated and controlled, but from the concept, it doesn't really matter. And what this pressure controller does, it periodically applies pressure. So it pumps in cell culture air, just 5% CO2, normal air, into the chip, and then pumps it back out. So it adds overpressure. And what the overpressure does, it pushes the medium to the satellite chambers, and then it releases the pressure and the medium flows back. And this has a couple of advantages. Maybe the biggest advantage, very practical advantage for those of you who might have already worked with microfluidics, there's never any problem with air bubble formation. So normally, as soon as one has a closed device, if there's an air bubble forming, which happens quite frequently in carbonate buffers, as soon as there's any gas bubble forming in these small micrometer-sized channels, that's essentially the end of the experiment. The channel will clog, and that's it. Whereas here, any air bubble that would form simply merges with this gas phase on top and is essentially gone. So there's never any clogging. We can run these chips for multiple weeks, have done this for years, and never had any problem with any clogging of any channels due to air bubbles. So it's a very practical thing, but nevertheless practically important. And then another feature that I would like to emphasize is that when you look closely here, one of the satellite chambers is connected to the center using a straight channel, the other one via a meander channel. And this means that if we are now pushing gas into the central compartment, there will be more of the liquid will flow into the straight channel because the resistance is proportionally lower, and less will flow to the meander channel. So this means that although we are only actuating the central chamber, we can nevertheless, by design, recapitulate differences in perfusion and flow rates that are the tissue-specific differences in perfusion and flow rates. So adipose tissue, for instance, much less perfused than skeletal muscle or liver, and therefore the adipose tissue would be behind such a meandered channel here, and therefore the perfusion rates are substantially lower. So we can control just by the design the stoichiometry of the flow. And so this is then how that looks very briefly. So now very soon it will show that there's now the gas pushed into the central chamber. And once the gas enters here, this pushes the media to the side. And then afterwards, this flows back, enabling a very smooth mass transfer and rapid mixing. So this is how that then looks in real time. Now we add a dye to one of the side chambers. And now once again, we press first in and then we apply under pressure. And this then results in a very gentle mixing of the dye in the device. We've now more recently also combined this with machine vision and real-time image analysis. So now we can, at each second in real time, evaluate the fill height of each one of those chambers. And this AI algorithm that evaluates these fill heights is very closely overlaps very closely with the theoretical predictions. So that works really well. And when we then put that into a model such as this COMSOL model here, as soon as we know the local concentration at any point in time in a given compartment, we can essentially use this by applying then diffusion coefficients and the mixing dynamics to calculate, to put it a bit provocatively, the concentrations of the analyte at every voxel in the chip at every point in time. So this can be directly fed in so that we better understand what is the actual mass transfer between the individual compartments. So this here would be an example of local insulin concentrations. And so now a biological example. So on islets are on one side. We can stimulate, as I briefly mentioned already, on chip with glucose. Glucose stimulates insulin release. insulin release is also happening at the physiologically relevant timescales. Then on the other side, the liver, primary liver spheroids, a couple of centimeters away. What we then see is when we now just change to the glucose, there's no insulin at all in the medium that we add. So any insulin comes from the islets on the chip. Then we see that if we increase glucose concentrations, postprandial levels, we see a repression of those gluconeogenetic insulin responsive genes. So really demonstrating a direct on-chip crosstalk between the two different tissues. And this can also be validated this year as one example by RNA sequencing. So we see that in high glucose upregulation of triglyceride biosynthesis, AKT phosphorylation, also that we validated by Western blot and so on. So overall, that allowed us to reconstitute a network of interactions. So glucose elevations result in insulin production. We then also see an increased ER folding load, unfolded protein response, ER stress. So all of those factors that have been reported in islets already many years ago at the molecular level as a response to hyperglycemia. The insulin, we can profile the insulin dynamics and kinetics. Insulin signals or activates, binds the insulin receptor in the liver, resulting in AKT phosphorylation and then further downstream, for instance, SREBP activation, which links it to de novo lipogenesis, and also a repression of gluconeogenesis, which in turn then is a negative feedback to circulating glucose levels. So this here was a two-tissue constellation. We're working on this more complex four-tissue scenario, where again we are interfacing this with a sampling system. So now 3 times a day we add a pulse of high glucose, postprandial glucose levels into the chip through the inlet. This would be the red curve here. So glucose gets injected into the device, gets distributed via pumping into the individual compartments. Then the pancreas realizes that now glucose levels are elevated, secretes insulin. The insulin gets distributed in the compartments, and then the glucose sink tissues are responding to the insulin by taking up more glucose, and thereby ameliorating these spikes to fluctuations within the physiological range. And in order to achieve this, we use a system called physiological scaling, which means that we are adding the right amount of each one of those tissue models to recapitulate the human function in vivo. So for instance, adding the right amount of islets so that the circulating insulin levels are indeed the relevant postprandial insulin levels. And by then starting from this healthy stoichiometry, we can also vary the stoichiometry, adding, for instance, more adipocytes to mimic aspects of obesity, for instance. We can also directly take patient-derived material, for instance, from diabetics that are already insulin resistant, in order to evaluate the effects of these different pathophysiological features on the overall maintenance of glycemic control. Then this would be disturbed. Now we are within the hyperglycemic range, and then we can ask, okay, which compounds could we now add in order to push that back down and restore glycemic control? And all that I've talked about here is glucose, but of course we can also add whatever other factors. We can add fructose, we can add different amino acids, various other supplements, of course also pharmacological agents. And also one pet project in the lab, if one wants to call it like this, integrate the microelectrode array here in the skeletal muscle in order to have different regimens of electrophysiological stimulation. So then we could ask, for instance, is it more beneficial to have high intensity or low intensity stimulation? Is it better to have it before the glycemic pulse or before the glucose pulse or after the glucose pulse? So all of these are possibilities of linking the different interactions. And since the system is accessible, we also sample every 2 hours. We sample 2 microliters out of the device after replenishing some of the media. And this can then also be used to interrogate or profile a variety of different biomarkers and also their kinetic profiles. So with this, I'm done. I think my time is also up. So I hope that I could convince you that these organotypic culture systems of primary human cells can indeed be cultured and recapitulate important aspects of steatosis, inflammation, and fibrosis. These patient-derived cultures can mimic the inter-individual variability and also the heterogeneity of human mesh. We can identify both established and novel targets for MASH in this context using chemogenomic screening. For TRPM8, this is a very interesting target for us there. We are using different MASH animal models now as well as develop new inhibitors. And in the future, we hope that these more integrated circuits can simultaneously emulate drug response and toxicity in a more integrated all human context. So with this, I would like to thank everyone in the lab in Stockholm at Karolinska, everyone in the lab in Germany, the Bosch Institute of Clinical Pharmacology, all the collaborators, and of course also all the funders, and would be more than happy to have answer any questions that any of the audience might have now. Thank you.
Thank you very much, Volker. Very impressive, very nice talk with a lot of information and data. Are there any questions?
I have a question.
Please go ahead.
Hello, Volker. Glad to see you again. Thank you so
Nice.
much for the great presentation and the insightful presentation. I have a question regarding your muscle D models. Have you performed in your laboratory some in vitro functional assays by exposing your muscle D models to hepatotoxic drugs or drugs taken by muscle D patients like statins, fibrate, antihypertensives, most frequently taken drugs by muscle D patients, by obesity and that things, to see the interplay between hepatotoxicity and muscle D, to see this interplay?
So we've done a huge hepatotoxicity screen, but this was indeed not in muscled cells. So we have not repeated that in muscle cells. So we've done in total 120 compounds, also with a concentration response series, 120 compounds, half of them with clear clinical indication for drug-induced liver injury, and the other half as very clean negative controls, all normalized for therapeutic exposure levels. So using Cmax and multiples of the Cmax. So in this non-patient derived context, we've done quite a lot of work, but we've never compared this directly to patient-derived cells or materials. It's an interesting question and could certainly be addressed.
Okay, thank you so much.
Maybe I can add on to this question. Did you check in your muscle D model statins, which has been indicated that it could be actually beneficial for muscle D patients? Do you have to look at that?
I don't believe statins were part of that. I would have to check. So we've tested a variety of different compounds. So I believe everything that was at present at the time of performing these studies, everything that was in phase 2 or phase 3 for MASH. I'm not aware that statins were indeed part of that, but I might be wrong there. So I would have to go back to the list to be sure.
Okay. Any other questions?
Yes. Hello.
Hello. Please go ahead, Leo.
Great presentation, really amazing work. It's very hard to do all that. Maybe a question about can you say something about the medium that you use to keep all these organotypic cultures happy?
Yeah, it's a very basal medium. And it's all published, and I would be also more than happy to send it to you. So it's a Williams E base.
Okay.
No serum, no growth factors, no cryptic morphogens or any of these nature. So it's a very basal medium. And importantly, this works, so that's at least my interpretation. This works because it's already fully differentiated primary cells. They don't have to proliferate, they don't have to differentiate, they are really locked into their molecular phenotypes and are going to stay there. So if you would use anything organoid, anything that is derived from stem cells, I would be quite certain that this would not work like this, because we always see that once we are withdrawing the differentiation factors, they are at least partially reverting their phenotype again. So the analogy that I always like to give to the students in the lab that also ask this question quite frequently is that in human there's also only one blood. So as unexpected as it is, so with classical cell lines, one is used for decades essentially that every cell line has some little tweaks and to maintain the cultures. But here it's really very, very basal, and all of those tissues work that we've tried so far.
Because just as a follow, muscle cells are usually not cultured in the same way as liver cells or fat, right? So
But there it's typically derived from myoblast. So what we are doing for the muscle is that we are micro dissecting out the actual bundles. So they're including the tendons. We just cut them entirely out. So we can do that from the mouse. Then we use the FDB muscles. We can also do it for human. Then we would use the intercostal or the jaw muscles. So, but it's also there, fully differentiated. And essentially we just have to treat the cells well enough so that they don't die. Yeah, but we don't have to drive the differentiation process anymore.
Right. Okay. Well, impressive.
Can you indicate which papers you were referring at, Volker? That you said which medium composition you were saying it was already published.
So that is in the initial characterization paper from 2016. There's already the medium is fully disclosed. Now for the muscled paper, as soon as we want to do metabolic manipulations, we vary glucose concentrations, of course, or insulin concentrations. So those can be varied in a wide range. You know, typically 1.7 micromolar is the classical medium composition, which of course is astronomical when it comes to insulin levels. So there we go to the normal ranges between 0.1 and 2 nanomolar rather, so 1,000-fold lower.
Right. Okay, thank you. Bernard?
Yes, thank you. Congratulations for your talk. I have a question about the part where you presented the results regarding the organocatalyst. Okay, and if I may have not understood, so my question might be silly. If I understood well, you found one organocatalyst that was able to reduce fibrosis on the MASH model.
So
right?
It was actually already a chemical series. So this, so the concept had been described before in the science paper that I also referenced on the slide.
Yeah.
And then based on this initial compound, there was a chemical series of similar chemical derivatives. And so it screened the chemical derivatives and then zoomed into the one that had the most effect in our MASH model.
And reducing fibrosis in this
Correct.
model. Okay, and so yeah. So my question is then, do you know whether this catalyst can repair the mitochondrial DNA or help to the repair of mitochondrial DNA? And so we might imagine that this compound might favor mitochondrial function and reduce the oxidative stress.
It's an excellent question. So part of these series that were developed after we've done our screen. So part of this series is actually directly mitochondrially targeted. So
okay.
I at present couldn't really answer your question, but it's a kind of very nice avenue and area to investigate further. Absolutely, I fully agree with you.
Okay, thank you.
So hopefully in 3-4 months, I should hopefully be able to answer this question.
Okay. Thank you.
Okay, Marie-Hélène.
Yeah, thank you. Thanks a lot for your presentation. It was very nice. I worked on myospheres, spheroids, and adipocytes spheroids, and I observed that cells didn't stay in spheroids. So did you look at that? Did you observe that cells were not always in spheroids and they were attached in the cell support during cell culture? And did you observe also another thing that I looked? It was that some cells can proliferate during cell culture, and others not. And it was not a problem for you.
So let me answer the second question first. So proliferation, indeed, we don't see at all. So, or if I say at all, there might be some very rudimentary baseline maybe. So if we do an EDU staining, we typically see that something like 2-3% of cells might have some form of cell cycle or DNA synthesis going on. So very basal. So this is for the cell proliferation. Having said that, we have actually developed and also published a cocktail of compounds that can drive very efficient cell cycle entry of primary human hepatocytes. So essentially that consists of a Wnt canonical GSK3 beta inhibitor, Wnt signaling agonist, TGF-beta inhibitor, and NF-kappa B activator. So those 3 compounds alone are sufficient to drive cell cycle re-entry of primary human hepatocytes in around 60% of cells. So that's for the proliferation part. But at baseline, everything that I showed you in the normal conditions, no proliferation. And this is neither for the hepatocytes nor for any of the other models. I must say, we've characterized the other models not in that much detail yet, but at least we haven't noticed anything. So, and then when it comes to cells detaching from the 3D culture, so interesting enough, we just had a meeting today in our lab meeting for culture of one specific donor, something we've never seen before. One specific donor where we saw some cells almost raining out, of the spheroid. We believe that this is a subset of immune cells, but this is very recent and very fresh from the press. We've worked with this now for 10 years. It's the first time that I've ever seen this before. So that's why if you had asked me yesterday, I would have said no. Now I say maybe it can happen sometimes. But it's a very, very rare occurrence. And if at all, it must be some specific subset of cells that happen to sneak into the supposedly hepatocyte isolation there. And for the other models, we've not seen it. So I think you said myospheres?
Yes.
So yeah, since
so-
we're using microdissected muscle bundles, they're longitudinal with the tendons attached on both sides. So there we've never seen anything detaching.
Okay, thank you.
Thank you.
Any other questions?
Yes.
Okay, go ahead, Bernard.
Yes, I'm sorry. I have another question about mitochondria because I am a little bit mitochondrial centered in my work. I just want to know whether you had the opportunity to measure mitochondrial function on your muscle spheroids, for example, with seals.
We are just setting up the COS assay, so it works. That I can already say.
Okay.
And we have a student that is working on this as we speak, but with muscle so far we have not tested it, but that should be something that is quite ready quite soon. So we know that we can profile overall mitochondrial activity. The profiles in the look good. We see that there's a modification or a change in the overall mitochondrial activity if we use menadione as a positive control. So this is what we can already say. And now we are testing that with other, be that mitochondrial toxins or other conditions that we are exposing the cells to.
And in the muscle condition, I think it will be very interesting with your model to make a kinetics. I mean, to see whether the mitochondrial function is varying depending on the time of the culture.
Absolutely.
Okay.
And also that would, of course, nicely link also to OGG1 modulation.
In addition to that, yes, interesting. Yeah.
Yeah, I think once, that's always a nice thing when such a new assay, at least for us, would be set up. There's many questions that one can ask. Is there differences between different donors? Is there differences between disease states? Is there difference? Also we have material from everything from F0 to F4. We have differences in NAS score.
Yeah.
So do we see any systematic trends there when we're comparing donors, or when we treat with different compounds, or when we change the nutritional composition of the media and therefore sort of emulate a nutritional intervention, if one wants to call it that way. So there's many indeed interesting questions to ask. It's a matter of paucity of hands.
Nice perspective. Yes, thank you.
Any other questions?
Get a comment, Michael.
Okay, yeah, please go ahead, Maribel.
It's impressive work, Volker, and it has been very, very nice and stimulating talk what you have given today. I wonder if in those microphysiological models that you have used with the fluidic devices to emulate drug response, if you could integrate the renal function, the renal tissue.
Yeah, that's a very, very good question. So all the tissues that I've shown you so far was where we can take purely primary cells. Yeah. The problem with kidney function is that is much, much harder because it's not only the cells themselves, but it's also the recapitulation of the actual anatomy. So we couldn't just isolate the cells and throw them all on a pile or make a spheroid out of it and then hope that this is going to be emulating renal function. So that's why we ourselves have not yet dared to touch the kidney. And the one option, which I think also from the literature looks really nice and promising, those renal organoids, at least some of those are really nice from what I see in the literature. The problem with those would be that I would be very skeptical that they will work in the same media for exactly the reasons that we discussed before. So once it's a stem cell-derived model, I would not think that those cells would be happy in the long term in basal media composition. So that's the main problem. I fully agree with you, and I would love to integrate it, but I think we are simply not there yet.
Yeah, but you're getting closer. And the other question that it always worried me, we have advanced a lot on hepatocellular damage, the hepatic level, but you have talked very early in your talk about cholestasis model. Where are we now at this moment regarding the cholestasis models?
So what we do for cholestasis is we are exposing the hepatocyte spheroids to a cocktail of physiologically relevant concentrations of a bile acid mixture. And what we then see is that when we are exposing them to the cocktail, there's no toxicity. Normal spheroids tolerate this just fine. When we add a hepatotoxin that is non-cholestatic, we don't see any amplification of toxicity. However, when we add a cholestatic compound, we see a drastic synergy between the bile acid cocktail and the toxicity of the cholestatic agent. So this is the model in a way that we can identify by this synergy between the bile acid mixture and the cholestatic compound, an amplification of toxicity for the screening or identification of cholestatic toxicity.
Yeah. So you are considering some problems with the transporters, biliary transporters.
That is most likely the underlying reason. We've not investigated that in detail for those compounds, but it's known that many of the compounds that we tested there are indeed BSEP inhibitors, for instance.
Yeah. Thank you. Congratulations, Volker.
Thank you very much.
Any other questions? Okay, then I think it's time to finish this seminar. Volker, once again, I would like to thank you for your outstanding lecture. It was a pleasure to have you with us, and looking forward to future interactions.
Absolutely. Thank you again for the very nice invitation. It was also a very pleasant discussion, very good questions, more suggestions also for future experiments. So I really enjoyed it. Thank you.
Yeah, Volker,
Thank you very much.
A very well attended meeting because we are in the middle of Easter time.
Thank you.
Okay.
Bye-bye.
Bye-bye.
Thank you, Volker.
Bye.
Bye-bye.
Bye-bye.